pleasure to welcome everyone here. Thank you for coming. We're here for Dustin Sokolowski's PhD defense. As you know, I'd like to thank the examiners and committee members for coming as well. And for so many people, Dustin's family is here as well. This is really, I think, maybe the first Toronto native that's given a PhD defense in my lab, so it really has a nice feel here. I'm really excited about this day today. I'm not going to say too many words, but I have to at least introduce Dustin and say a couple of things. So I got to first meet Dustin through Anna Goldenberg. So Dustin was a summer student with Anna, and I was asked to go to a meeting about some exciting new tool that Anna's lab has developed, and then there was Dustin here, who was an undergrad student at the time. Anna showed something, or one of her postdocs showed something, and I was like, oh, that's interesting. What about this? And then, I say that all the time. And then 3 minutes later, Dustin goes, oh, well, that's this. And then this is basically in real time was analyzing things, checking stuff out, giving feedback. And I was like, who is this guy? And it wasn't the first time. And that kept, even in that meeting, it happened a few times. I'm like, this is good. And then Anna was super kind enough to say, Mike, you should think about Dustin. Dustin's interested. I think he's a good fit for molecular genetics, where they're using computational biology to answer biological questions. And so Anna gently suggested that I consider this. And so I was lucky enough to have Dustin rotate in the lab. And he obviously came, which is fantastic. We also retained Anna as part of the committee, so this has been really wonderful. He's been attending my lab meeting, Anna's lab meeting. He's made great contributions to both labs. He's actually getting a training award today at the same time as his defense, and thankfully, Wayne is going to actually speak on my behalf at Roy Thompson Hall to introduce Dustin, who's not going to be there. And so a couple of things, since you're not going to be there either probably, I'll say a couple of the things that I'm going to ask Wayne to say that I said, and Anna's echoed as well. So like a lot of great trainings, Dustin has published some nice work, and he's also, he's got scholarships, and this is wonderful. At the same time, he's been super generous and really had an influence in the lab. His can-do attitude isn't just for projects that he's involved in or that I really care about. He's like that with everyone in the lab, but also people on the floor and also people across the world. And so Dustin was a very different student for me, where we started with one really clear project that I'm still super focused on and interested in. And then Dustin's working away hard. He's like, oh, there's a problem here, and I want to come up with a new idea to solve it. So that's chapter one. It wasn't what I was thinking of, it was Dustin's idea, and given his interest and his passion for this, I was happy to support him and do the best I can to help with that. But really, so the work you're going to see today is coming. These are basically Dustin's side projects. This is kind of, in a way. So from my perspective, but the side projects have become a really helpful part of my own lab. So I'm also I like to adapt and learn from my trainees as well. So he's actually shaped the lab and the work of others. So it's a really nice kind of feedback that I've been witnessing I had the privilege to be part of in real time, which is really nice. The stuff you won't get to see today, which is things that I was really keen about, getting Dustin really interested in comparative genomics and looking between species genomes. He's actually done that on the side. You won't hear about it today, but with him and some of the people in the lab and Melissa Holmes' lab, we've got the new naked rat genome. And we're starting to look at its wonderful attributes, and Dustin's coming up with new ideas of this every day. You're not going to get to hear any of that, so you'll have to talk to him your drinks after another day. But just to say that kind of attitude, the really generous nature, the willingness to help others and to improve science, and Dustin's passionate about open science, and when doing something that's of good quality matters a lot to me, and we might be a bit slow, but I like to say what we do is good and used by others. Dustin's taken that to the next level, which I've been very happy about, and it reflects in how people interact with things that he's created. So I want to put any more pressure, but I know I'm speaking on behalf of Anna as well. We both nominated him for the award, but I think it's well deserved, and he's been, it's his day today. I'm not going to take any more of your time, Dustin, but take it away. I wish you all the best in your exam. Looking forward to your talk.
Thank you for the very kind words, and for everyone for coming. Wow, this is a real mix of people in my life. That's really great. Thank you so much. So today I'm going to be talking about my PhD work, which I entitled Applying Transcriptomics to Unveil the Underlying Biology of Complex Traits. And hopefully by the end of the talk, this title and what I've done to try and address this will make a lot of sense. Sorry, just want to make sure the slide, okay, we're changing slides with that mouse, got it. So first I want to talk about what is a complex trait from our perspective, from a biologist's perspective. The textbook example that you'll see is something like height. It's typically quantitative in nature, And the variability in this trait, so for example, how tall you can be, is dependent on the variability of many genetic factors. In fact, for height, there have been over 5 million people's genomes who've been analyzed in the context of height, and there have been 12,000 unique genomic regions associated with it. These traits are also, the variability is controlled for by environmental factors. For example, my grandfather, all of his sons and I are around 5'10" to 6'2", and he had size 13 feet, but because he grew up in the Depression, he only ended up being about 5'6" tall, which is likely due to effects of nutrition. And of course, the interplay between your genetics and environment. So in this toy example, one allele that you have might make you more sensitive, or might make your height more sensitive to nutrition, while with another allele, your height might be less sensitive to nutrition. And those of us who study these complex traits in a genome-wide context would know that the variability in this genetic sequence doesn't always account for a huge amount of the variability of the trait. And this is sometimes called the missing heritability problem. So us in molecular genetics like to take a gene regulation approach to this. So DNA is not just a naked sequence, but there's a massive amount of factors regulating how and where a gene might be expressed, including the accessibility of the DNA. DNA is really tightly wound, so being able to get at the gene, the proteins that bind to, and the small molecules that bind to the DNA to impact transcription, alternative splicing and polyadenylation to impact what and how stable the RNA molecule is, and also RNA modification, so small molecules binding to the RNA themselves, and of course not including everything that can happen at the protein level. And each of these regulatory factors are variable on the cell type that you're looking at, the tissue that you're looking at, the stage of development in disease, and of course your environmental exposure. And now we think of these modes of gene regulation as how the environment interacts with your genome to predict a complex trait. So thanks to the work of thousands of experimental biologists over the last half century, we're able to measure many of these mechanisms of gene regulation and gene expression itself at a genome-wide level. And for my PhD, I'm going to be talking about RNA sequencing. So RNA sequencing measures the abundance of RNA for every gene within the transcriptome of a sample. I think it's sort of a really great first approach for hypothesis generation and looking at complex traits, as it's the first readout of the genome, being it makes RNA. It's very flexible and multiplexable, meaning that you can look at lots of individuals or experimental groups at a time, and you can also look at different classes of RNA molecules. You don't need to know the sequence beforehand, it's sequence independent. And lastly, compared to other genomic approaches, it's quite cost effective. So, there are data sets where you can look at thousands of samples individually. Two types of analysis that are very common with RNA sequencing are differential expression. So you look at the average differences in gene expression. So for example, each point in this volcano plot is a gene between your two groups, say taller individuals and shorter individuals, and you can have a coefficient of how much this gene is changing in expression and a measure of significance, and also co-expression analysis. So here, imagine you have a population of lots of people with variable heights, and you could find genes whose expression patterns change together and are associated with height. And typically, these genes that change with each other are under a shared regulatory, or are under a shared biological regulation. So I've sort of talked more about this bulk RNA sequencing side, where you have conditions that you're interested in, say you're a smaller and larger animal, a tissue that you're interested in. You measure all of the RNA in that tissue, and you can get an average gene expression between all of your cells and compare between groups. But this RNA sequencing technique has been extended further into single-cell RNA sequencing. So here, every individual cell is shot into a little tray or grid, and there's a small amount of RNA sequencing that is completed on each individual cell, which is barcoded so we can keep track of it. And so you can look at the different cell type populations that are in your sample, as well as the gene expression, or some measures of gene expression of every single cell in your sample. Of course, I'm not the first person to notice that RNA sequencing is valuable and interesting. This is a plot showing the exponential growth of publicly available RNA sequencing data. And as of now, there's about $50 billion worth of experimental data that's publicly available, meaning that you and I could just download it right now if we wanted to. So, as you can probably tell, I'm not an experimental biologist. I'm coming at this from the lens of computational biology. And analyzing RNA sequencing from a computational biology or bioinformatics lens can be a very interesting problem. So each sample can have a range of tens of thousands to hundreds of millions of unique measures, meaning RNA, across tens of thousands to hundreds of thousands of features, being genes or expressed regions. Different experimental designs can have a huge variation in sample numbers. You know, if you're trying to annotate a genome, you might only need 2 individuals with different tissues. Whereas you can look at population level RNA sequencing data sets where there's thousands and thousands of samples. And because of this, there's also a huge number of experimental designs and hypotheses that you can ask using RNA-seq data. Lastly, once you have all of this information, the output is actually quite similar. It's typically a list of genes that are associated with the complex trait or the hypothesis that you're asking. And converting that list of genes into something that's biologically interpretable, or in the case of some people at kids who use RNA sequencing in the clinic, translational, can be very, very challenging. So there's 3 common questions to RNA sequencing analysis that I'm going to talk about today. And each one of these questions are divided into a different chapter for me trying to answer these questions. So the first is, are the changes in gene expression that we see driven by changes in regulation or changes in cell type composition between our samples? Secondly, how can I publicly use this massive treasure trove of publicly available RNA sequencing data to better understand my results? And lastly, are the transcriptomic dynamics that I see driving the trait? So are these genes that are changing impacting height, or a byproduct of the trait? Are these individual at different heights and therefore the genes are changing in expression? So again, each one of these questions is subdivided into a different chapter, and I'll start at the very beginning, which is a tool that I developed called Single Cell Mapper, which aims to take bulk differentially expressed genes and assign cell type specificity to them. So let's sort of review that idea. You have a basic bulk RNA sequencing experiment. People in Mogen have seen something like this many times from each other's talks. You have some experimental condition, a tissue that you're interested in. I like the hypothalamus, and we'll be talking about that later. And this tissue has lots of heterogeneous cell types that all can do different things. In bulk RNA sequencing, you take all of the RNA you can from all of these cell types and put them into a homogeneous pool. And then you sequence, so randomly sample RNA molecules from this pool, and you enumerate them. Then with differential gene expression, you can compare the average gene expression in that tissue between groups. So, you know, gene 1 might have a 20% increase in the larger mouse, and we can assign some statistical significance to that. With single cell RNA sequencing, you can identify cell type specific differentially expressed genes within a sample. So this is sort of a fairly common type of plot. This is a sort of mode of dimensionality reduction. Each point is a different cell, and each cell has RNA sequencing completed on it. And so based on the gene expression similarities between each cell, we can cluster cells together and say that these cells, for example, are likely from the same cell type and or cell state. And then we can compare the average gene expression of these genes to all of the other cells that we've measured in the single cell RNA sequencing data set to measure cell type specificity. We can then integrate single cell RNA sequencing or cell type specific gene expression with this bulk RNA sequencing data to estimate the cell type proportions in your data using a class of methods called RNA-seq deconvolution. So there's a very long list of different ways that you can go about this, but all of these approaches effectively try and measure the proportion of variation in your bulk sample that's accounted for by the variation in your cell type specific gene expression. And that amount of variation is very strongly associated with the amount of each cell type that's in your bulk population. And with Single Cell Mapper, what I'm trying to do is to link cell type specific gene expression and cell type specific proportion to infer the cell type specific origin of differential expression. Like I said, cell type specific about 10 times there. So, how can we look at this cell type contribution of differentially expressed gene? Well, the cell type contribution is some function of the specificity of that gene, so what cell type the gene likes to be expressed in, and the cell type proportion, how much of that cell type is in your bulk sample. But if you think about sort of that RNA sequencing experiment, well, there's sort of this randomization of RNA molecules that get into this homogeneous pool, and then another random sampling of RNA molecules from the pool to enumerate our count matrix. So from a sort of biological and RNA sequencing technical perspective, these measures of cell type specificity and cell type proportion are independent because we're randomly sampling at every step. Computationally, however, it's not quite as simple. So I've sort of gone through these methods, but what you might have noticed is that the cell type specific measurement, or sort of this measure of cell type specificity, is also used in calculating cell type proportions. So much like our dog Lucy, these values computationally are not independent from one another. So to address this, I introduced this normalization step that's based on leave one out orthogonalization, where for every differentially expressed gene I'm interested in, I remove it from the cell type specific gene expression matrix, and I remove it from my bulk data, and I recalculate cell type proportions conditional on that gene. And so now, since these values have been computationally orthogonalized, I can reweight the fold changes of the bulk RNA sequencing data that you're inputting into this by the cell type specificity of single cell RNA sequencing, which could be your data or publicly sourced, as well as by the cell type proportions across all of your samples from this integrated deconvolution, and by the inverse ratio of cell type proportions between your two experimental groups. Because I'm not interested in, well, I'm trying to differentiate, I should say, differences in cell type composition and cell type specific gene expression. So if you have a gene that likes to live in a specific cell type, and then that cell type infiltrates your treated sample, then we actually didn't find a difference in gene expression, but we found a difference in like cell mobility and infiltration, which has different biological interpretations. With these cell-weighted fold change metrics, you can use single cell mapper to identify which cell types are driving your differentially expressed gene. So here's a toy example where we have the red gene here, the cell-weighted fold change calculated for each gene using single cell mapper. So you have a measure of cell type specific gene expression and all the cell types. By using a very simple outlier cutoff recalculated for every single gene, single cell mapper can assign these differentially expressed genes to the cell types, to their cell types of origins if they're above the cutoff, or sort of predict that the differential expression that you detect is unlikely to be from these other cell types. And similarly, if you have a fairly homogeneous set of cell-weighted fold changes, none of them will pass this outlier cutoff, and therefore your differential expression is from some averaging of all the cell types. You know, maybe some housekeeping gene is increasing in expression. So I sort of evaluated this tool mathematically in the simulations and with a handful of data sets. But I thought for time, I could show one example that might be more intuitive from a user's perspective. So there was a study that came out in 2020, Devallier et al., and they looked at gene expression of kidney regeneration over multiple time points. And I really liked this study because they had the bulk RNA sequencing data, they had lots of additional experimental validations, and they also had single cell RNA sequencing data. So I could sort of see how single cell mapper did at every step of the way through their validations. So I took this data set and looked at the first time point, reprocessed the data and calculated differentially expressed genes in the first 3 days of kidney regeneration. I then took a publicly available kidney single cell RNA sequencing data set and pushed these through single cell mapper to try and find cell type specific gene expression changes in these bulk data. So this is sort of what a standard output of single cell mapper looks like. You get a heat map, or in this case, the columns are genes, the rows are cell types, and genes in orange are where you're likely to have some cell type specificity. And of course, it also returns like the actual cell types each gene is assigned to. So the first thing we saw was this signal of fibroblasts and endothelial cells, which the original authors also saw. But I was really excited to see that using single cell mapper, we were able to see this very large immune response. This was exciting because the authors did not report this finding in their RNA sequencing data, but due to their sort of additional experiments, this immune cell response was basically the whole point of their paper. So had single cell mapper been out at the time, their sort of like figure one first thing they did could have sort of led to their final point that they found. So as I mentioned in part of their validation, they sorted for immune cells, T cells in this case, naive T cells, and they looked at differentially expressed genes just in those T cells between kidney regeneration. So I took that data, reprocessed it as well, recalculated differentially expressed genes, and I asked how well do my immune assigned differentially expressed genes from their bulk data match up with their cell type specific, like true cell type specific differentially expressed genes. found a highly significant overlap, and of the 81 overlapping differentially expressed genes, 80 of them were changing in the same direction. So this was very encouraging. So yeah, single cell mapper has been out for a couple of years. It's a fully maintained CRAN package, so it should be relatively easy to use. And one of the things that I really liked about having a package and method that's sort of accessible and gives relatively intuitive results is that I've gotten to see how other people can use single cell mapper and even adapt it to their own hypotheses. So I've gotten to see, learn about a lot of biology I would have otherwise missed, just because it's not my field. So moving forward, the first thing I would say is for a single cell mapper, 2.0, it'd be great to be able to incorporate cell type specific differential gene expression. When I started building single cell mapper, it was extraordinarily difficult to compare different samples at a single cell resolution, look at differentially expressed genes. Now this, I would say, has been upgraded from extraordinarily difficult to very difficult. But there have been tools like Bayes Prism that came out last year that are being, and Bayesian mind algorithm that are able to incorporate sort of average differences in gene expression across single cell samples to infer cell type specificity of a bulk RNA sequencing sample. Secondly, single cell omics has moved into the world of single cell chromatin accessibility and single cell DNA methylation. So I didn't get into it in this talk, but estimating cell type proportions of bulk RNA sequencing data is very, very variable based on the method that you use, the hyperparameters you might tune, and even whether or not you're using single cell omics data or immunohistochemistry data or large public consortium. And this is much worse for single cell accessibility and methylation. But once that problem is solved, I think that single cell mapper is actually very well suited for these data types. So with differential expression calculated, an idea of which cell types these genes came from, I asked, how can I interpret these genes and how can I use this currently existing literature to best help my data interpretation? And to do that, I built a tool called DEEP, not the insecticide, but the differential expression enrichment tool to build a set of differentially expressed genes that are really homogeneously curated and that you can interact with to better interpret your data. So in a perfect world, we're back at our complex trait height. We have a tissue that we're interested in, and let's say we find 1,000 differentially expressed genes between my groups. If all of the literature that had RNA sequencing data was perfectly curated in a list, I could take each one of these differentially expressed genes and assign them to studies that interrogate the individual components, individual biological components that combine to make my complex trait. So in this case, the effects of hormones, the effects of cell differentiation, and the effects of diets on height. And so by sort of sorting these genes, I can see how each gene is impacting all of the biological processes that eventually make up my complex trait. But we can't do that, at least not yet. And it's not due to the inaccessibility of publicly available RNA sequencing data. I hope I sort of made that clear. But of course, I should say publicly available data has been used quite a lot to assign genes to functions. Loads of people have processed these publicly available RNA sequencing data to generate novel discoveries. And you saw about 3 slides ago, these data are often reprocessed to benchmark novel methods. So RNA sequencing reanalysis and meta-analysis can be quite challenging from a technical perspective. Of course, how you design the experiment and how you do the experiment is fixed and variable across every lab and every experiment and even the time of day you do the experiment at. But there's also a huge amount of variability in the data analysis portion. So for each step of RNA sequencing analysis, there's maybe a dozen different totally acceptable tools that you could use to analyze the data that way. So if you think about all of the combinations of how these tools could be used, there's literally thousands of perfectly acceptable bioinformatic pipelines to process RNA sequencing data. And while these pipelines are all accessible, they can have a pretty substantial impact on results. So one way to circumvent this issue is to take that many groups have sort of tried to do, including the Leak Lab, or the Jeff Leak group, the Jeff Leak group, was to sort of in huge batches reanalyze all of this publicly available RNA sequencing data. So they used sort of a combination of cloud computing and really clever alignment to repeat the quality control, alignment, and quantification of 70,000 human RNA sequencing samples, including 2 very large, very well annotated consortia, and a few hundred studies, so about 50,000 human RNA sequencing studies from sort of smaller groups. And where I went into Recount 2 and tried to redo as much of the differential expression as possible. So you might be asking, it seems like they did the hard part, which, they did. But why didn't they continue and redo all the differential expression? And the answer to that is because while the sequencing data is consistent, the metadata related to these samples are very inconsistent. So very briefly, metadata is really just any information that can help explain what the sample is. From what condition is it, to what tissue is it from, what sex is it from. And public metadata can be very inconsistent and outright missing. So Shannon Ellis, who's a postdoc in the Leak Lab, a year later looked at these 50,000 samples and saw that only about 9% of them had sex annotated. And these are all human samples. So it's not like, there's no sex assigned to a tree or something. And in fact, there was one study she found where the sex was labeled as Donkey Kong and Bowser. I guess someone thought they were funny and forgot to switch the labels. But using RNA sequencing data, some of this metadata can be quite accurately predicted. So thinking of that sex example again, if you find genes that really strongly map to the Y chromosome, you can say with pretty high confidence that the sample came from a male. So this is what she did. Training on the Genotype Tissue Expression Project, which has human RNA sequencing data from 10,000 samples across, 1,000-ish individuals spanning 10,000 samples and dozens of tissues. She built a machine learning classifier based on a series of linear regressions to predict common metadata in the Cancer Genome Atlas and Sequencing Read Archive. So she sort of validated this model with TCGA and then predicted metadata in these remaining 50,000 samples. So I now have the bulk RNA sequencing data, and I had metadata that was reported. I collected metadata from many sources, and we now have this predicted metadata to fill in the gap. So with help from the super talented undergrad who I worked with for a couple of years, Jed and On, we were able to do a series of data imputations, transformations, and component analyses to make this mixed metadata consistent enough to be able to push it through a large differential expression pipeline. And that's exactly what I did. I used DESeq2 as my differential expression tool, and pushed through thousands of gene lists and then annotated them afterwards based on how biologically interpretable the gene lists seem to be. And then I also, for each of these lists, enriched biological pathways that are associated with these gene sets and transcription factor gene pairs. When the dust settled, I was able to have about 3,142 sets of differentially expressed genes. Most of them still come from these very large, very well annotated data sets. Again, this is because of sort of the really fantastic metadata elements. But I still included 142 unique studies spanning about 150 tissues and cell lines and almost 1,000 different biological conditions. Of course, this is somewhat biased by what people like to study and what people are funded to study in human RNA sequencing. So if you input a gene list that is related to, say, cancer, DEET's going to output more studies that are related to your gene list than if you, say, input a list of some obscure parasite infecting some obscure cell type. But I really do think there's at least something for everyone. From a user's perspective, all you have to do is input a list of genes, human gene symbols, preferably with a p-value and coefficient, but not required. And DEET will output a set of comparisons and their associated studies with significantly overlapping results to yours, as well as comparisons whose shared differentially expressed genes are correlated. So meaning that not only are these genes overlapping, but they're changing in expression in similar directions to you, which adds an additional layer of confidence that there's some shared underlying biology here. And DEET also completes a series of visualizations for you so that in a perfect world, you can run DEET, take a figure, and stick it in your paper. So again, there are a decent number of validation approaches that I used to make sure DEET seemed accurate, and that it was also representative of sort of a wide share of biology. But from a user's perspective, I think this one's probably the most intuitive. So these are all publicly available data sets that are in DEET, right? So many studies report their own differentially expressed genes. So if I want DEET to work as a data, literature review tool to help people interpret their data, I should be able to take the original study's differentially expressed genes, input them into DEET, and DEET would output my version of the differentially expressed genes from the same study. So I input this Gormally et al. Differentially expressed genes into DEET, and DEET spits out all of the comparisons from Gormally et al. That I reprocessed. So I did this across 35 comparisons, including studies in the SRA, TCGA, and GTEx, and this all checked out fine. So we felt pretty confident that DEET was working well enough for people to use and for their own data interpretation. Yeah, so I'm going to talk about a use case in the next chapter where I used it for my own work. So for now, DEET also available and in use. It was published earlier this year, so I haven't really gotten to see how people use DEET yet. But I built a Shiny app with Kai, so that, who's in the back, so that you don't need any programming experience to be able to use DEET. And people are using it every day, so hopefully sooner or later some interesting things will come out of it. Yeah, so moving forward, I think the biggest hurdle by quite a long shot is scaling this metadata. So as compute power increases and people have realized that these large reprocessing efforts are really valuable, groups like Recount has updated, and new groups like ArcGS4 have come in and have reprocessed millions of samples across many species, which also includes more biologically controlled experiments in animal models. If you asked me how long it took to process all that metadata for the 70,000 samples, I'd probably say about 2 years. And if I had to do it again with all the lessons I would learned, I would say it'd take me a year and a half. So this is really not scaling. Again, I'm not the first person to notice this. Groups like BioStudies are sort of full dedicated teams to consistent metadata curation and RNA sequencing data. There are software tools that exist that try and predict metadata, not just from the RNA sequencing data, but also by sort of imputing missing metadata within the metadata itself. And if I were to start DEET 2.0 tomorrow, I would want to take a community science approach. So I think that people want their data to be accessible and used, and for their papers to be sort of understood and appreciated. And every one of these publicly available data sets has a corresponding author and the information for the bioinformatician who submitted all this data. So if I were to make a web portal where they get an email, the drop-down of their study shows up, they can fill in any missing information, and importantly say, this is the experimental variable, this is what we're stratifying for, this is what we're blocking for. That information could be instructions that are fed directly into DEET 2.0, and then we could have this passive increase of reprocessed differentially expressed genes. That is sort of, in a sense, as scalable as people are willing to contribute. Yeah, so we have our differentially expressed genes, we have the cell types that they're coming from, hopefully, and we can use this publicly available data to better interpret our results. And for my last chapter, I applied these tools as well as other state-of-the-art bioinformatic approaches to the complex trait that I'm interested in, which is puberty. So why puberty? First of all, we all go through it. Maybe some of us more gracefully than others. And puberty is sort of a complex biological process and a complex trait. So from a biological standpoint, puberty is considered to start in the hypothalamus, where naturally pulsating gonadotropin-releasing hormone increases quite dramatically in frequency and amplitude and is released into the pituitary gland, where luteinizing and follicle-stimulating hormone is secreted into the gonads, where the hormones that I'm sure everyone in this room recognizes, testosterone, estrogen, and progesterone, are cycled back into the body, creating this positive feedback of pubertal activation. The hypothalamus is this really interesting tissue. It's the tissue that's responsible for the connection between the endocrine system, so our hormonal control, and the nervous system. And a big part of that is through these awesome cells called neuroendocrine cells. So they input signals from neurotransmitters, just like a neuron, and they interpret those signals, but rather than interpreting the signal and passing it along to the next neuron, they also secrete hormones in response. So from the context of a complex trait, we like to think of puberty, to make it, quantitative pubertal timing, so when an individual goes through puberty. The timing of puberty is highly heritable. Of course, this biological regulatory mechanism is hugely conserved across mammals and rodents, including the sort of specific hormones in the timing. We've looked, or I guess other groups have looked at the association between pubertal timing and genetic variants across hundreds of thousands of humans, and have found hundreds of associations, but just like height, there's not a huge amount of heritability that these associations are explained for. And pubertal timing is very, very environmentally sensitive. So in humans, we think of puberty as a function of age, which is fair, it's sort of the main function. But your pubertal timing can be strongly influenced by your nutrition, your sex and gender, by aggressive or social interactions with other individuals, by larger social networks, and even by things like your circadian rhythms. So from the Wilson lab, where we really like studying gene regulation, puberty serves as this model to study gene regulation and plasticity in a non-disease context. But if you are interested in translational medicine, pubertal timing is very important there as well, as improper pubertal timing, traditionally defined as puberty that occurs 2 standard deviations too early for your population or too late, has direct effects on some very severe later in life health outcomes like cancer, cardiovascular disease, mental health disorders, and bone disorders, and diabetes. So of course, we took a transcriptomics approach to this, or else it wouldn't be in my thesis, where we took the hypothalamus from the black 6 mice and spanned 5 different time points before puberty, during puberty, and after puberty in males and females. And this experiment was done with, I guess, the original puberty group here at Sick Kids by a really, really awesome team, and I was able to inherit this fantastic project. So from those of us who like to do RNA sequencing, you might be thinking, wow, 50 samples is a lot, and it is. But thanks to Kyoko Yuki, a former postdoc in the lab, she was able to build a protocol for a robot, she was able to build a protocol for a robot that allowed for this automated library preparation of RNA sequencing data, so that we can compare all of these 50 samples to each other simultaneously without worrying about batch effects. So the first thing I did was differential expression. I looked at differentially expressed genes between each pairwise time point in males and females individually. And we saw some sort of patterns that we somewhat expected to see. So before puberty, we see that the hypothalamus is the most transcriptionally dynamic. And this is related to gene groups related to cellular growth, which makes sense. The mouse brain about doubles in volume at this time, as well as this decrease in transcriptional repression, which is known as a hallmark of pubertal activation in the hypothalamus. We also see this substantial increase in myelination, which is a phenotype that has been observed in humans, sort of is increase in myelination from birth to late adolescence in many tissues, but has not yet been observed in the hypothalamus, so that's sort of exciting to see. During puberty, we see this hormonal activation. So there's not many genes that change in expression, but nearly all of them either encode peptide hormones in the hypothalamus or encode regulators of these peptide hormones. And then after puberty, we see a decrease in hormonal activation and an increase in transcriptional repression, which I interpreted as this sort of return to baseline now that this sort of crazy pubertal network is completed. I then wanted to look to see which one of these differentially expressed genes are associated with differences in pubertal timing in humans. So I overlapped my differentially expressed genes with genes assigned to puberty through an Age of Menarche and voice-breaking GWAS studies, and found that in fact it was the differentially expressed genes well before and well after puberty that were most strongly associated with variations in pubertal timing at the DNA sequence level. So I thought this was sort of interesting. So I took these differentially expressed genes, I pushed them through DEEP, which was my chapter 2, consistent set of differentially expressed genes in human studies, and I found that the top hit was this set of differentially expressed genes between infant and child males in the dorsolateral prefrontal cortex, and my aging hypothalamus, or I guess developing hypothalamus differentially expressed genes. And these genes, the ones that are highly correlated and differentially expressed in the same direction, predominantly map to pathways that are involved in neurogenesis and neuronal organization. So while we set out to study puberty, this big transcriptomic signature is really this cross-tissue, cross-species conserved signal of neuronal growth and organization that occurs before puberty. And it's sort of interesting to see that these are also the genes whose variation can impact pubertal timing in humans. But not really what we were looking for. We're here to study puberty. So we went back to the drawing board a little bit and tried to see how could I take all of these time points in both sexes to capture a signature related to puberty. So starting with a great suggestion from Gary Bader, realized that I could use puberty, the difference in pubertal timing in males and females, both in mice and in humans, as a feature to study pubertal activation. So the idea here is that since males and females go through puberty in a sort of different times, we should be looking for genes that, as you go through developmental time, have this start diverging in gene expression patterns in males and females. Computationally, this translates to looking for age by sex interactions of gene expression in our mice. So there's a number of ways that one could do this, and I ended up picking this approach that leverages a very max rotated principal component analysis. So very two-sentence overview of PCA. Principal component analysis is a dimensionality reduction technique where you can take lots of correlated genes and shrink their variance into an individual vector. And what can be very helpful with PCA is that this vector of sort of control, genes where their variation is shrunk, is orthogonal to this vector, which can be very helpful from an initial data quality control standpoint. However, this orthogonality has this cost where we don't necessarily know how the principal components relate to individual samples. So the varimax rotation overcomes that constraint by rotating your principal components in nth-dimensional space until each principal component aligns with your samples. You can then look at a varimax principal component score, which is literally what coordinate each animal is at on the principal component, to look for age by sex interactions on these principal components. And then you can look at principal component loadings to see which genes are associated with these age by sex interactions. So I performed this analysis across every one of the rotated principal components I computed and found one rotated PC with an age by sex interaction across development. So the x-axis here is each time point we measured. The colors are female and male mice, and the y-axis is the rotated PC score. I should mention that the sign of the rotated principal components is arbitrary, so genes can also have the flipped on this 0 line here pattern of gene expression. And then I wanted to look at genes, of course. What are the genes associated with this age by sex interaction? And I found that the 4 genes with the top loadings, so the most associated with this age by sex interaction of all the 15,000 genes I measured, were all genes that encode hormones in the hypothalamus which have been previously associated with pubertal regulation. So this was very encouraging, and also let us sort of confidently look at the rest of these genes, thinking that they are at least associated with this pubertal signature. And generally, the remaining genes that were part of this age by sex interaction were involved in synaptic signaling, myelination again, and hormone activity, of course. So I was interested in looking at the cell type specificity of these age by sex associated genes. So I took a publicly available single cell RNA sequencing data set that happened to have day 14, so prepubertal, and day 45, postpubertal mice. I took these genes and inputted them into single cell mapper, my tool for identifying cell type specificity, and found, first the thing I found was what we sort of expected to see, which was this substantial hypothalamic neuron and neuroendocrine enrichment of these genes. Many of these genes, like promelanin concentrating hormone and oxytocin and vasopressin, have been associated with puberty before. But there's also some relatively new genes, which I don't have time to talk about, but you can read the paper, or its preprint now, or the thesis. But we also still saw the signature of oligodendrocytes, which is related to that myelination thing I was excited about earlier. So the job of an oligodendrocyte is to myelinate neurons. So neurons have this electrical impulse going along their axons, and by myelinating neurons, the axons are insulated, and the signal can jump along these nodes and can really improve and facilitate neuronal signaling. So we see this sort of cell type specific gene expression pattern in these age by sex associated genes, but we also see an increase in cell type proportion. So before puberty, you can see this sort of increase in my mature oligodendrocytes. This is using RNA-seq convolution. Increase in mature oligodendrocytes sort of up into the middle of puberty, and then you see the inverse relationship in oligodendrocyte precursor cells. So you see this expansion from precursors to mature oligodendrocytes. I then wanted to see if I could find age by sex associated genes that are associated with this oligodendrocyte expansion that seems to be occurring from before to about the middle of puberty. The single cell RNA-seq data set had this sort of beautiful lineage of oligodendrocyte precursor cells into developing oligodendrocytes through to mature oligodendrocytes. I know lineage tracing can sometimes be a little bit unreliable, but for this lineage in particular, every step has been very well characterized. So I'm sort of pretty confident in it from a biological perspective, which let me use a tool called Slingshot to computationally trace this lineage, and a tool called TraitSeq to identify genes, so each row here, whose expression patterns, more expressed in red, are associated with this oligodendrocyte expansion. So these are the 58 age by sex associated genes that are associated with oligodendrocyte expansion. This is statistically significant. The first thing that I saw were, again, more genes that have been previously implicated in puberty, both from a neuronal perspective and from a neuron-glial interaction perspective. And secondly, I saw these other genes that are associated with like fundamental oligodendrocyte expansion. And I thought it was sort of interesting to see that these genes had this age by sex interaction of gene expression in my data, as it suggests that perhaps the oligodendrocyte expansion itself is playing some role in puberty, maybe playing a sort of facilitative role, allowing neurons to interact sort of in more dense ways at a crucial time. So yeah, in conclusion, I found genes that are associated with pubertal development rather than just its related growth phenotypes. While I wasn't able to talk about it today, I found new neuronal candidates involved in pubertal development, and a cell type, these oligodendrocytes, previously unassociated with puberty in the other than that we've seen oligodendrocytes expand in other tissues. Again, I'm a big fan of open science, so with Kadia, who's also in the back, we built a Shiny app so that if you're interested in your favorite gene in the hypothalamus, you can just type it in and see how that gene changes in expression along postnatal development. We also have it for the pituitary gland, which Kadia focuses on. So, moving forward, my initial thought was to sort of talk about, well, you know, gene expression is just one measure of gene regulation, and there's all these other modes, and we should be looking at that. But if you've been to talks in Mogen before, you've probably heard that many, many, many times. So I thought I would maybe switch it up a little bit, and instead talk about how one could maybe meta-analyze all of the models of pubertal regulation that exist. So our lab has started to introduce, and this paper is sort of really trying to hit home this age by sex model of pubertal development, where you take a wider window and incorporate age. But also, if you feed mice with a consistently high-fat diet, they go through precocious puberty. Or of course, if you gonadectomize them, they will never go through puberty. Similarly, using genetic models of puberty, for example, taking an inherently leptin-deficient mouse, you can raise them until after they would naturally go through puberty, treat them with leptin, and they'll occur a vaginal opening within 2 or 3 days. And finally, if you're feeling a little extra brave, you can go into the non-model organism space and look at species whose pubertal activation is somewhat unrelated to age. So if you've talked to me before, I've probably talked your ear off about the naked mole rat, but their pubertal activation is sort of suppressed through psychosocial interactions within the colony rather than from developmental time. And so I think if you were to sort of look at all these models together, and I think what's called a dandelion plot, you could sort of parse out what aspects of puberty are sort of being driven by the biology underlying these models. You know, sort of like remove age, remove this sort of metabolism aspect, that sort of thing, remove sex hormones. So, in conclusion, I hope I've convinced you that gene expression and regulation is a really crucial avenue to study complex traits. Though even with a pretty tight experimental design and, you know, inbred mice, it can be quite challenging to go from the actual experiment to interpretable biology. I hope that with single cell mapper and DEET, I've pushed the needle forward a little bit to help interpret these experiments. And looking at my complex trait, puberty, I was able to incorporate age by sex, age and sex into the same model to find a really nice pattern of pubertal regulation. So that's it for the talk. I do have some acknowledgements if you are willing to bear with me. And the first person I would like to acknowledge, of course, is Mike. We don't have a picture together, so thank you for showing me. Yeah, as Mike sort of mentioned, we met when I was still an undergrad, about 21 years old, with like full head of steam, truly bright-eyed and bushy-tailed with all of these ideas of things that I wanted to do and didn't really understand. And you have found this way to foster, a critical eye on the data that we have, sort of a realistic approach to how to do experiments, how to do data analysis, how to ask questions, without dampening my enthusiasm for science at all, which I think is a really amazing balance that not very many people could strike. And also, I think what you did with me was very brave from a supervisor perspective. You have a big lab, you're a grad chair, you have lots of things going on, and I was in here saying, I don't know if we can look at the cells that well. You mind if I spend 18 months doing this other thing that neither of us thought about that much? And you sort of were both willing to do it, and you learned with me, and we were able to publish something that was genuinely very valuable for people. And was like we meant to do it the whole time. So I've loved being in your lab, and really do consider you a friend and mentor. And thank you. Anna as well. Anna's been with me since, I guess, third year undergrad. And you have been this absolute rock through everything, whether it's sort of work-wise or just in my personal life. I can't count the number of times where I've come to you with something feeling very anxious, and you see through all the noise and go like, no, just do this. And, even things like, I don't know what to do for grad school, my undergrad program doesn't really have this mix, just go talk to Mike, it's fine. And really made my grad school experience so much more smooth, both from the work and from decisions. And of course, your lab has been amazing to work with all of these years. Our work has sort of diverged a little bit from the genomic side to the clinical side of it over the years, but the sort of underlying core, how do we look at data, how do we know what's real or an artifact, has been absolutely invaluable for the last 8 years now. Of course, my committee, Anna and Mike I mentioned, but also Phil, thank you for coming to every single committee meeting, always having really great ideas, and I guess preventing us from getting lost in the sauce. We have been very involved in every step of this along the way, and you've really kept us on track. And of course, also to Jesse and Kathy, thank you for taking the time to read my 240-page thesis and come and talk to me about it. And Jesse, also thank you so much with DEEP and with single cell analysis over the years. It's been really great learning from you. And of course, Sarah, thank you for coming all the way from Washington to come and talk to me. It's been really, really great. So I wish I had time to talk about everyone in the lab, but I'm going to sort of focus on 3 people where my thesis experience would have been fundamentally different without them. The first is Lauren Erdman, who's on vacation, very well-earned vacation in Cancun right now, but still took the time to Zoom with me when I was worried about how to interpret something for my defense. She's so generous. I have been with her since I was a second-year undergrad in Paul Arnold's lab on the 12th floor, who's not here anymore. Lauren told me to come with her to Anna, and I could keep helping out. Lauren was there when I met Mike the first time, and has been there every single step of the way, just so unbelievably supportive and helpful. Brian, you are sort of fun. You really are, I think, the captain of the dry lab of this ship. You've been extraordinarily patient with me from coming in when out of the 4th year of my undergrad thinking I totally know what I'm doing, to helping me learn how to truly be a strong computational biologist. There is so many things that I do where I know it's probably fine because it's how you do it. And Mariela, of course, from early conversations of talking about, well, what is this data analysis that we're missing, that eventually became, single cell mapper, to, of course, introducing me to endocrinology and into mole rats, into what has shaped a huge amount of my PhD and also likely my postdoc with Jared. Yeah, I cannot imagine what my PhD would have been without you either. And as well, of course, the Wilson Lab. Spent a ton of time with you guys. It's a really, really amazing environment. Pretty much every time I'm in, I'll scoot my chair over to Ryan or Katie or Mariela or someone and ask some question, and within 5 or 10 minutes I see that everybody is slowly congregated because the conversation was interesting. And I just love the enthusiasm for science. I love that we can learn about super diverse biological things because we all study very different things, but have that fundamental understanding of how to do genomics, how to evaluate an experiment, and we can sort of, I think, help each other grow. And we get to hang out together a lot, which is great. Also, Catherine, who I think is still here, might be at the RI retreat. Yeah, I think if our lab is a ship, she's the duct tape. Mike and I aren't like the most organized people in the world, and she has really been there to keep us on track and bail us out countless times. And as Mike mentioned, a huge part of, a huge gift of working at SigKids and this institution is having access to really wonderful collaborations. These are just some of the people who I've gotten to work with during my PhD, as well as Jared, who I've worked with through my PhD and will be continuing on with a postdoc. I think science really does have this intersection between job and hobby, and the ideas that we have and the experiments that we design can be quite personal, and especially in the genomics world, data collected is very hard won. So thank you for trusting me with your ideas and with your data. I never took that responsibility lightly. Also, I guess sort of a shout out to the Center for Computational Medicine team. I installed like an insane amount of data, or an insane number of packages just to try with them and play with data. And every time for the SigKids network, I would get in touch with them and they would do all this stuff, and then I would try the tool and go, it's too much effort, and then try something else. And they were like really great and patient with me. I know it's their job, but they knew me. And also thank you, of course, to all of the funding sources to make any of this possible. Yeah. So one lovely thing about being in grad school for this long is that I've gotten to make friends through grad school. There's Laura, who's on Zoom, who's in Vancouver now. She introduced me to my sort of new favorite hobby, rock climbing. She's introduced me to my favorite person, Jacqueline. I've gone from rolling myself over to see what she's doing across the hall to traveling 3,000 kilometers to seeing her, and it's always awesome. Jan, thank you for letting me live in your apartment for the last 4 years. For always being there to talk. And I don't have many pictures with like everybody else I met through grad school, sorry Lucas. But it's been really nice having a group of people where I can talk about more technical stuff and in the same conversation switch to pure nonsense or music. It's been sort of a really great way to blow off steam and stay engaged. And also the friends I've had through my whole life. Nikki, you've been my sibling for a long time now. I feel like we've been sort of standing shoulder to shoulder for nearly 2 decades, and you've really played a huge part in who I am today and are really inspiration of who I want to be. Raj and Victor, who's in China, you two are also people who I lived with during undergrad when I wasn't a jam. And you guys are really two of the most thoughtful people I've ever met in my entire life. Laura likes to say that boys are friends shoulder to shoulder, like we stare at a thing together. And I will say something sort of in passing, not remember I said it, and then 4 months later you'll go, how's that thing going, with everybody's name and the specific context. And of course, everyone else, like Liz and everyone who I've been friends with since elementary school, primary school, who I've still talked to today. It's been great being friends with you guys from all stages of puberty. Penultimately, my family. It's like amazing. My 90-year-old Bobby is here in the crowd. It's been lovely talking to you basically whenever I'm in a car about like, interpersonal relationships with work. It's better to hear strong speakers. About the work itself, I love talking to you. Zadie, who can't be here with us, he really sparked my love of animals and nature, and that is at this point what I want to spend the rest of my life doing, learning about the creative and interesting things that animals can do. Of course, Joel and Toby, my uncle and aunt, who are here and have always been this like incredible, steadfast line of support for me. And these guys. Yeah, Jeff, my brother-in-law, doesn't feel like a brother-in-law. Has been around since I was 11 years old, and we went to Western together and rock climbed together and played squash together, and is truly a brother. Mariah, I know we always joke about who's the golden child, but you have been this like shining beacon for the next hour. Shining beacon of positivity in my life, and You've never, ever cast a shadow in any way. You're so wonderful and make everybody feel like they're part of it. Like, truly, I feel like your successes are mine, and I'm so happy to be there. Dad, you, sorry. Yeah, the story I like to tell about my dad is that in the 80s, during the AIDS epidemic, there were very few people who were there for people. And he was one of the only dentists in Toronto who were willing to take on patients, despite having no idea the risks of anything. And like, you have that sort of strong sense of right and wrong sort of to your core. But I also know that you care about us a lot. And so being able to talk to you and knowing that you're able to balance really what's right and what's right for us is amazing. And Mom, I don't know what I can say about you that I didn't write in a full protected piece about you. But yeah, I mean, we didn't really know like anything other than that you were our mom and that science is cool until embarrassingly late, probably. You have always been there for us, exactly who we need at every stage of our lives, whether it's our mom, a mentor, a friend. You know, I've heard a lot in the halls or in conferences of people saying like, I don't know if I can do science and have a family. And I've always been like, no, it works. You know? And so thank you for being, you know, everything. And last, but certainly not least, Jacqueline. Yeah, there's a lot of wonderful and challenging things that I've had to experience through my time in molecular genetics and that I've met through molecular genetics. And you are by far the most wonderful and most challenging of all of them. I can't wait to move in with you next week.
Thank you very much, Dustin. I think for the sake of time, we're going to have to forego questions. The committee cannot ask questions, probably for the best of Dustin, that he doesn't get into a fight before his defense. Thank you so much, Dustin, and for sharing really thoughtful impressions of your time here. It's a great seminar. Thanks everyone for coming. The examiners, please do stay here if you want to take just a bio break here. The exam will start actually at 2, so it starts now officially. And so if I can kindly ask everyone to leave.